Hello everyone. Uh, Today we will talk about epigenetics and comparison of the genetic inheritance with epigenetic inheritance. So we already know about the genetic inheritance, that these are the genes that are transferred, the inheritance that is transmitted normally through your genetic code. That's what we call genetic inheritance. And in addition to genetic inheritance, we also have epigenetic inheritance. The epigenetics means the study of changes caused in organisms caused by modification of gene expression rather than the alteration of the genetic code itself. So in this case, the genes remain the same. There are no mutations in the genetic code. Rather, it's the expression of the genes is altered because of the reasons other than the mutations. For example, methylation. So methylation can certainly have impacts on the expression of the genes, and these patterns of methylation can also be transmitted from one generation to the other generation. For example, we have here we see the methylation of DNA itself, and generally these are the cytosine residues or the cytosines which get methylated in case of DNA, and generally the cytosines which are followed by a guanine. And we call them CpG islands. So basically P stands for the phosphate group, and this dinucleotide here, it means we call them CpG. CpG means that the C is at the 5' end and G is at the 3' end. And both of these two nucleotides are connected via a phosphate group. So these cytosines often get methylated in your genome. On the right side, we see and these regions where these dinucleotides are found, we call them CpG sites, and a bigger region is known as CpG island. So as you can see here are multiple cytosines followed by guanine. So these are our CpG sites, and most of these sites can get methylated, and this is called as the DNA methylation. Another methylation that can take place and have an effect is the methylation of the histone proteins that we have already talked about. So what is the difference between the two types of inheritance is that in case of genetic inheritance, for example, we have a gene X here. This is switched on, and something goes wrong with this gene, and there's some sort of mutation, and the DNA sequence is changed, and this changed or this modification is passed on to the next generation when the cell duplicates itself. So even after the multiplication of the somatic cells, this gene X remains switched off. And then in the production of germ cells, this also remains switched off when germ cells are produced from the somatic cells. While in case of epigenetic inheritance, this is a little different. For example, here we have gene Y, which is switched on. There is some sort of chromatin modification. So here we are talking about the methylation, for example, or some sort of modification here, which is not in the genetic code. So there's nothing wrong with the genetic code. Rather, it is some sort of modification in the gene expression, and no alterations in the genetic code itself. So which leads to this modification leads to the turning off of gene Y, not because of any modifications in the DNA, rather some other modification, not in the genetic code itself. So A, T, C, Gs, etc., they remain the same. During the multiplication of the somatic cells, they remain switched off, but when production of the germ cells takes place, this can temporarily change the pattern of methylation, and this can lead to the switching off of gene Y. So it's not a permanent alteration in this case during the production of the germ cells. Let's see how could this affect the expression of certain genes. here we use a term what we call the Histone code. So the Histone code is basically a methylation or acetylation code. So this is the methylation or acetylation pattern on different histone tails. Here's an example of H3, a histone tail. And we can see that we have this arginine, lysine, and serine residues, and some of them can get methylated, some of them can get both methylated or acetylated, and there are some other which can get phosphorylated. So not all of them would be methylated or acetylated or phosphorylated at the same time. Rather, these patterns may change depending upon the different situations. And what would that mean? For example, here we have certain examples. On the left side we have this modification state, and on the right side we have the meanings. Well, what does this thing mean? So if we look at the modification state, for example, if the lysine residue at 9th position, the 9th residue, if this one is methylated, this would lead to heterochromatin formation or gene silencing. So you do not need to modify all of them like this. So these are the chances of these residues that they may get methylated or acetylated or phosphorylated, but you won't see this pattern on any histone tails at any given time. So this would be different. Not all of them would be either methylated or acetylated at the same time. So here the lysine at 9th position is methylated. If this one is methylated, so that's the code. If this, and in this code, sometimes even modification on a single on a single amino acid may have an impact. It may have a drastic impact. So here in this case, for example, the modification on 9th residue, the lysine, if this one gets methylated, this can lead to heterochromatin formation or gene silencing. On the other hand, if the lysine at 9th position is acetylated and the lysine at 4th position is methylated, that would lead to gene expression. That would be, if this is the pattern on the tail, that means the gene would be expressed. And once again, if serine at the 10th residue and the 14th residue lysine, if they are phosphorylated and acetylated respectively, then this would lead to gene expression. And another important thing, what we call silencing of the X chromosome or the X chromosome inactivation. You know about the X chromosome inactivation. This generally takes place in the females, because females have got 2 X chromosomes and one of them needs to be randomly inactivated, especially in mammals. Although there are certain examples where there's some paternal preference. I mean, the X chromosome from the father would be preferably inactivated. But generally it's random in mammals. In human beings it's random. So one of these 2 X chromosomes needs to be inactivated so that the expression of the genes which are located on the X chromosomes is not duplicated. This is also known as sometimes known as dosage compensation, that one of the X chromosomes needs to be inactivated. And this generally X chromosome inactivation takes place at early blastocyst stage. So the default status of inactivation, the default status we can say is inactivation of the X chromosomes, and one of them stays active and the other one stays inactive. Although there are also studies that which say that 12 to 20% of the genes on inactive X chromosome can also remain expressed. So it's not completely silenced. But in any case, in this example, we talk about the X chromosome inactivation. So on histone 3, in case of X chromosome, the lysine residue at 27th, the lysine residue at 27th position, this needs to be methylated, and this single methylation can lead to chromosome inactivation, and that may also be true for silencing of the Hox genes. We have already talked about the Hox genes. So basically, the patterns of DNA methylation are, so we have talked about two things. One was DNA methylation. The second one is the methylation of the histone proteins. Both of them have the ability to silence the genes, and both of these two patterns can also be transmitted. But the methylation patterns of the DNA are relatively more stable in somatic cells, while the patterns of histone methylation can change rapidly during the course of the cell cycle. Despite this difference, several studies have indicated that DNA methylation and histone methylation at certain positions are connected. Uh, in an interestingly coordinated process, proteins that bind to methylated DNA also form complexes with the proteins involved in deacetylation of the histone proteins. So we have a scenario where DNA is wrapped around these histone proteins. We have previously discussed this. For example, here, the DNA is wrapped around the histone protein, so modifications are taking place at two different places. There is methylation on the DNA, and there is methylation on the histone tails. So both of these two methylations, the methylation on the DNA and the methylation on the histone tails, they can be coordinated. That when a protein that binds to the methylated DNA, it can also bind with the proteins which are involved in the deacetylation process. I mean, here you have got these acetyl groups. So if the DNA is methylated, a protein will come here, bind to the methylated DNA, and remove the acetyl group. And we have previously studied that deacetylation can lead to heterochromatin formation. Acetylation relaxes the chromatin, while deacetylation leads to heterochromatin formation, or it condenses the chromatin. So if you have a methylated DNA, then a protein can bind to methylated DNA and then remove the acetyl group. So it means you have a dual impact. So one thing, this methylation of the DNA is going to have a negative impact on the expression of the genes. It would not let the genes expressed. You might have studied about the CpG islands generally, or a methylation of the promoter regions. So promoter regions where there's a higher number of these CpG sites present. If these promoters get methylated, they may also lead to the suppression of gene expression, and that's the reason that when your DNA is methylated, proteins can bind to the methylated DNA here, and they can remove the acetyl groups. When deacetylation takes place, when these acetyl groups are removed, then this may lead to the compaction of the chromatin. And when chromatin is compacted, then this would inhibit the expression of the genes. And so this is the reason that when DNA is methylated, nearby histones are deacetylated, resulting in compounded inhibitory effect on the transcription. And likewise, a demethylated DNA, when this is demethylated, there's no methylation on the DNA, this does not attract deacetylating enzymes to the histones. So DNA is not methylated, then it will not recruit those proteins which are involved in deacetylation of the histone tails. And when deacetylation does not take place, then these histone tails remain acetylated, and when they remain acetylated, they are more mobile, and this promotes the transcriptional activity. In most cases, methylation of the DNA is fairly a long-term stable conversion, but in some cases, such as in germ cells, as we have seen here in germ cells, while silencing of the imprinted genes must be reversed, here is reversion taking place. Here the genes are silenced, and these genes need to be switched on. Then demethylation can take place to allow for epigenetic reprogramming. The exact mechanism for demethylation are not entirely understood. However, it appears that this process may be mediated by the removal of the amino groups by DNA deaminases. After deamination, the DNA has a mismatch and is repaired, causing it to become demethylated. We have studied today that other things, some other modifications of the chromatin or of the DNA can also have an impact on the switching on or switching off of the genes. These do not necessarily need to be mutations in the genes or in their promoter regions to silence them. Rather, methylation can also lead to the silencing of the genes. And we have discussed today that there are two types of methylations. One is the methylation that takes place on the DNA in the CpG islands, while the other methylation is the one that takes place on the histone tails. And both of these two methylations can have impact on the expression of the genes. That's what we call epigenetic inheritance, that there would be a specific pattern, a specific histone code on the proteins, and both the methylation code on the DNA as well as the methylation code on the histone proteins, both of these two patterns can be transmitted to the next generation. The only difference here would be that the modifications in the DNA, when they take place, they are more kind of permanent, while methylation patterns, they are not so permanent, and the genes can be switched on by removing the methyl groups and temporarily allowing the genes to be switched on when germ cells are formed, and that would also be true for the X chromosomes. So I think that is sufficient for today. Thank you very much.